Hello students, we are here today and this is lecture 45, the last topic of your unit 5 and also of your course Pharmacology-II BP 503T. Today we will be talking about bioassay of digitalis, histamine and 5-hydroxytryptamine. I am Dr. Monica Sachdeva, Professor at Rajkumar Goyal Institute of Technology, Ghaziabad. So, during my lecture, this presentation, we will be talking about what is digitalis, what is its role, where it is used and how the bioassay of digitalis is carried out. We will be also talking about histamine, on which type of receptors histamine acts, what is the role of histamine and how bioassay of histamine is carried out. Then we will also talk about bioassay of 5-hydroxytryptamine, what is its role and how bioassay is carried out of this 5-hydroxytryptamine or serotonin. So, let us start with bioassay of digitalis. So, before moving on to the bioassay of digitalis, let us first talk about digitalis. It's such a beautiful plant. It is called as Digitalis purpurea. The biological source is the leaves of the Digitalis purpurea, and the common name of this plant is foxglove. We know that digitalis, it is a cardiac glycoside. The active constituents from digitalis, they are digoxin and digitoxin. It is a cardiotonic and it is used in the treatment of congestive heart failure by sodium potassium ATPase pump. Now, to carry out bioassay of digitalis, the principle remains the same, that we have to have a test sample and a standard preparation. Now, since it is a cardiac drug, so we will determine the action on the cardiac muscles of both the standard sample of digitalis as well as the test sample of digitalis. So, the principle is that the potency of the test sample is compared with that of the standard preparation by determining the action on the cardiac muscle. Now, what are the standard preparations? The standard preparation, it is a mixture of dried and powdered digitalis leaf. 1 unit, it contains 76 milligram of the sample. And how are these extracts prepared? Exact amount of the powder, it is extracted with dehydrated alcohol in a continuous extraction apparatus for 6 hours. And the final extract, it should contain 10 ml, that is 5 ml alcohol plus 5 ml water per 10 gram of digitalis powder and stored at a temperature between 5 degrees centigrade to minus 5 degrees centigrade. Similarly, the test samples and the standard solutions are also prepared. The standard and the test sample extracts, which we had seen earlier, they are diluted with the normal saline. Now, 1 gram of digitalis powder, it is diluted to 80 ml. So, this is how the same will be the preparation of the test sample, and the same procedure will be followed for the preparation of your standard solution. Now, to carry out bioassay of digitalis, we use 3 methods here. One is the guinea pig model, the pigeon model and the cat model. This method, it uses the endpoint method, or we talked about the quantile bioassay method. So, let us see how this guinea pig model, by this endpoint method we carry out bioassay of digitalis using guinea pig. Now, guinea pig is anesthetized with a suitable anesthetic agent. Jugular vein is traced out. Various adhering tissues are removed. It is cannulated by means of venous cannula. Now, a pin is inserted in the heart it is inserted in such a way that it gets inserted at the apex of the heart. Now, apex में लगाने का क्या advantage होगा? Whenever there is the movement of the heart जब भी beat करेगा, we can see the movement of the pin, up and down movement of the pin. So that is why that pin is inserted at the apex of the heart. Now this injection is continued through venous cannula until the heart it is arrested or it stops in systole. Systole means contraction of the heart. So when the systole की position में जब heart stop कर जाता है, तब तक we continue with the injection of the sample. Now the amount of the extract required to produce this effect is taken as a lethal dose of the extract, and similarly the lethal dose of the test sample is also determined, and we for both the test and the sample minimum 6 guinea pig they are used, and all the animals they should be of the similar strain. So the potency of the test sample it is calculated in relation to that of the standard preparation by dividing the lethal dose of the sample to the test, and it is expressed as the units per gram. So we just administer the test sample, we administer the standard sample and see at which dose the heart stops in the systole. So when that dose at which the heart stops in the systole, that is taken at the lethal dose, and this lethal dose by comparing of the test, by comparing it with the standard, we can find out the potency of the test sample. Now the cat model, similarly the cat model works on the same principle. The cat model is again the adult cats are used, it is anesthetized with chlorellos, the blood pressure is recorded. Now the drug is infused into the animal. Now the moment the heart stops beating and the blood pressure we record is 0, the volume which is infused, that is then noted down. Fine. So we have the two groups of animals, one is with the standard and one is with the sample. So कितना volume of fluid use हुआ heart stop करने में test sample के साथ में और standard sample के साथ में, we take those that is compared, and that is how the potency of your test sample can be calculated. So this can be calculated as threshold dose of the standard divided by threshold dose of the test multiplied by the concentration of your standard sample. So in this again we are seeing that cat maybe we are seeing when the heart stops and the blood pressure falls to 0, that the volume which we have injected for the test and the sample, they are compared, and the potency of the test in comparison to standard is then calculated. Then the other method we use for Digitalis bioassay, that is the pigeon method. Now in this pigeon method, minimum 6 pigeons are used for testing each sample. There has to be no evidence of any disease. The weight of the heaviest pigeon, it should not exceed twice the weight of the lightest pigeon. Okay. For example, if you have one pigeon which weighs 1 kg, so जो next जो pigeon होगा 2 kg से ज़्यादा का नहीं होना चाहिए. Then the food is withheld 16 to 28 hours before the experiments. The pigeons, they are divided on the basis of their sex, weight and breed. We divide them into 2 groups. They are anesthetized with anesthetic ether. Then one side of the wing is dissected. There is a alar vein. It is cannulated by means of venous cannula. Various dilutions, they are made with normal saline. Again the lethal dose is determined. Now what is the lethal dose? How do we find out the lethal dose? The stoppage of the heart. Now in pigeon, whenever there is a stoppage of a heart, there is a characteristic vomiting response which is called as the emesis response. So that stoppage of the heart or that vomiting response is taken as the endpoint, or in pigeons there is a crop sac from which milk is ejected out whenever the जो lethal dose होगा, जब heart stop कर जाएगा, जो lethal dose होगा, there is also milk from the crop sac of the pigeon that is ejected out, and that is also taken as a endpoint in response to the digitalis. So with the standard and with the test sample, by comparing both the volume at which this emesis or the ejection of milk is reported, that is taken as the lethal dose, and the concentration or potency of your test sample that can be found out in comparison to the standard. So for digitalis, we have used guinea pig method, we have used cat method, and we have used pigeon method, but the lethal dose is calculated whenever the heart stops. In guinea pig, the stoppage of the heart in systole; in cats, stoppage of the heart, and also when the blood pressure falls to 0, and in the pigeon, whenever their heart stops, there is a vomiting response which is called as emesis, or milk is ejected from the crop sac, that is taken as a lethal dose, and we can carry out the potency of the test sample in comparison to the standard sample. That is how the bioassay of digitalis is carried out. Now we move on to the bioassay of histamine. Now what is this histamine? We all know that जब भी allergic reaction है, there is the histamine is released. How this is caused and how we can carry out the bioassay, we'll just talk about. Histamine, it is a small water soluble molecule, or we can say it is an amine, autocoid, or a locally acting hormone, and it mediates its effects by binding to various receptors which we call as histamine receptors, H1, H2, H3 or H4 receptors. H1 receptors, histamine receptors, they are G protein coupled receptors, and they produce their effect through your IP3 DAG pathway. Now histamine is synthesized and stored at the following sites. They are synthesized in the neurons, enterochromaffin cells in the gastric mucosa, and most importantly in the mast cells. Now there are some other non-mast cell sources also where histamine is present in the body. They are also present in the brain, also sometimes functions as neurotransmitter. Then they are also present in the enterochromaffin cells in the stomach, where it stimulates HCl secretion by the parietal cells of the stomach. That is why when we give the antihistaminics, they are also used as the antacids. Now how is this release of histamine from mast cell is carried out? They are two types. One is the immune mediated and another one is the non-immune mediated. So what is this immune mediated? Now the first time when an allergic, it could be anything from which the person is allergic to, we call that as allergen. Whenever the person it comes in contact with that allergen, there is a large amount of antibodies, or we call immunoglobulin E antibodies, are generated. Now these immunoglobulin E molecules, they attach to the mast cells, but when the second time the person again comes in contact with that same allergen, these immunoglobulin E primed mast cells, they release granules and powerful chemical mediators which we call as histamine or we call as cytokines, and these chemical mediators, they are responsible for causing allergy, the various symptoms of allergy. Now the release of histamine, it is also carried out from the mast cell by non-immune mediated reactions. We have certain drugs जैसे morphine है, tuberculorin है, we know that they cause allergy, but they do not mediate it through mast cells or by immune reactions. They simply displace histamine from the mast cells. So this type of exposure, it does not allergic, this type of response, it does not require any prior exposure. And the pharmacological actions of histamine, so it depends upon the tissue, the type of the receptor which is present in the area of the release. So we have various receptors H1, H2, H3 and H4. Allergic reactions, they are mediated through H1 receptors. Now to carry out bioassay of histamine, we can record contractions of guinea pig ileum. Again, it is by a graded method, and we can also carry it out by a quantal method, that is fall in blood pressure in anesthetized cat or dog. It can also be used to carry out bioassay of histamine. Now to carry out bioassay of histamine using guinea pig ileum, we will first talk about this. In our first lecture, we had talked about that earlier lecture that guinea pig is very sensitive to histamine, to allergic reactions. So this animal, it is a specific animal, a very sensitive animal. So we are using guinea pig here. Guinea pig ileum, it is a very useful preparation for this bioassay of histamine, very sensitive to histamine, and it produces the contractile response due to the presence of H1 receptors in the ileum. And bioassay of ileum, it could be carried out on isolated guinea pig ileum by any of the methods we had discussed earlier. We could use matching method, we could use bracketing method, interpolation bioassay method, or multiple point bioassay method, that is 3 point, 4 point, 6 point, 8 point. Any of the bioassay method can be used to perform bioassay of histamine by using guinea pig ileum. So we have talked about this in great detail in our earlier lectures. You can see here that this is the setup of the assembly. This is the chymograph. We have a paper attached with it. Then this is the organ bath. This is the organ bath. This is the organ bath where you put in the tissue and your physiological salt solution. It also comes into this organ bath. Okay, and then this is the, which we call it as also as the organ bath, and here we call it as the tissue bath. This organ bath mein we fill the water, maintain it at a temperature of 37 degrees centigrade, and that is the liver is used, frontal writing liver is used, 1 gram load is used, the tissue is attached to this writing liver, और जब हम इसमें drugs डालेंगे, we put in the drugs into this organ tube, और जब भी हम drug डालेंगे, जो भी contraction होगा, that response is recorded here. And this response could be by bracketing, by any of the method you can perform this bioassay. And the physiological solution which we use here is the thyroid solution. और जो standard solution जो हम use करते हैं guinea pig का, that for guinea pig ileum, that is 10 microgram per ml solution. Now we will talk about the bioassay of serotonin, that is 5-hydroxytryptamine. Now this 5-hydroxy, what is this 5-hydroxytryptamine? It is a biologically active local hormone which has got a very low molecular weight and is a very important neurotransmitter found in the brain as well as in the periphery. And this was first detected in the serum, that is why called as serotonin. Thereby then it was also found in enterochromaffin cells of the gut mucosa. We call it as enteramine, and later on it was identified that both of these chemicals, which have been found in the serum as well as in the gut mucosa, they are similar, and it was called as 5-hydroxytryptamine, or we also call it as serotonin. Now, most of this, around 90% of this content of 5-hydroxytryptamine, that is localized in the intestine, and some of it is also found in the platelets, and it is also found in the brain. Then in certain stings, wasp sting, scorpion sting, 5-hydroxytryptamine is also present. Then certain invertebrates in plants like banana, pear, pineapple, etc., serotonin is also found. Now, this serotonin, it has got various receptors, 5-HT1, 2, 3, 4, there are 14 subtypes of these receptors, and they are again G protein coupled receptors and act through cyclic AMP pathway. And it has got various roles on cardiovascular system, in migraine, and in various other pathophysiological roles they are played by 5-hydroxytryptamine. Now, to carry out the bioassay of 5-hydroxytryptamine, again we have two samples, the standard and the test, and we can carry out, or we can carry out the assay in the biological samples by the isolated fundus strip of the rat stomach, or we can also use isolated colon of rat uterus could be used, or perfused rabbit could be used, but the most important method which is used to carry out bioassay of serotonin, it is by using isolated fundus strip of rat stomach. So, we will see how this is carried out. Again, it is a graded point bioassay. You can carry it out by any of the method, by bracketing, by interpolation, or by multiple point bioassay. The important thing here is how to dissect out the rat fundus. Now, rat fundus preparation, it is again a very sensitive tissue for this bioassay of 5-hydroxytryptamine, and this fundus strip, that is a very slow contracting muscle, and you have to now identify the fundus part of the stomach. It can be identified by its gray color, and it is situated above the pink colored thick pyloric part. So, we dissect out that fundus part and we cut the zigzag preparation of that fundal strip. And why do we carry out that zigzag? क्यों काटते हैं हम उसको? It is prepared so as to expose maximum proportion of the tissue to the drug. So, जो surface area है वो increase हो जाता है. That is why we prepare the zigzag preparation of this fundal strip. Now, for this we require Krebs solution prepare करेंगे, physiological salt solution. Standard और test के sample लेंगे, which is 1 milligram per ml. Stock solution. We prepare different concentrations by dilution method से various samples are produced. Now, what is the procedure? We fast the rat for 24 hours by carotid bleeding, by giving anesthesia, or by the stunning on the head. This method is not approved by CPCSCA now, so stunning method is not used. We give anesthesia, and by carotid bleeding we kill the rat or by sacrifice the rat. Then we fix the animal on the dissecting board. We open the abdominal cavity by horizontal cut in the middle region of the abdomen, identify the stomach, separate it from the abdomen by cutting the दोनों cardiac end और जो pyloric end है उसको cut कर देते हैं. It is placed in the Krebs solution और उसमें से fundus part जो है भी जो grey part है stomach में उसको हम अलग कर देते हैं. And then that fundus part जो है उसको हम ओपन करते हैं. I will show it to you in the diagram. This is how this is the fundus part. This is then cut out from this. इस dotted line पे इसको cut कर दिया जाता है और ये जो dotted lines हैं then इसको cut करने से this zigzag fundal preparation is then prepared. This fundal preparation is then attached to your writing lever, and the bioassay is carried out by any of the method, which it could be bracketing method, it could be interpolation bioassay, or it could be any multiple point bioassay. Here we use the Krebs solution as physiological salt solution. We maintain the proper temperature, we give aeration, pH is also maintained. So that is all about your bioassay of 5-HT of histamine. Thank you so much. I hope you have enjoyed the lectures. So this we come to an end of this pharmacology course PP 503T. Thank you.